traveled some great distance to get here. We're really glad to host you, to have you here.
There's a microphone.
So let me welcome you again. Especially those of you who came so far. We know that it's not easy for you, and hopefully you'll be awake this morning for a little while. You may get tired towards the end of the day, but we're really glad to have you here. I am in the Department of Molecular Integrative Physiology, and I actually got my graduate degree here. So I've been here for a number of years, and really am very committed to this university. And so we're really glad to have you here. Today I'm going to talk a little bit about cancer, and how many of you have had a biology course over the period of your educational experience? Oh, that's good. How many of you are engineer, bioengineer types? Okay, so anybody had any physiology course? Oh, good, we have some. All right, so since we have such a breadth of knowledge here, I want to try to explain so that everybody understands some of the basics about cancer biology. And a lot of the slides that I'm going to use today are provided by the National Cancer Institute. So if we think about cancer, it's really a formidable problem. If you look at the number of men and women who have cancer, every year there are nearly 750,000 men who are diagnosed with cancer, slightly less than that in terms of women. And if you look at, rather than talk about all of these, I want to try to highlight what I'll call the big three today. The highest incidence and the highest deaths caused by three cancers. Actually four, because one is male specific, one is female specific. And if you look in men, the highest incidence comes from prostate cancer. That's a hormone-driven cancer. Lung cancer and colon cancer then follow. If you look at women, breast cancer is the highest incidence, and this is a hormone-driven cancer as well. The lung cancer and colon cancer then follow. So this is terms of incidence. But then if we look at death rates that are caused, or deaths that are caused by cancer, we see a slightly different picture. We still have the big three, but now you see that lung cancer tops both of the profiles. Lung cancer is significantly more deadly in men and women, and it's a significant problem in terms of both health and in terms of cost. So if we talk about cancer today, if you were to think of what is involved in cancer, you said you had some biology? A little bit, okay. So you ought to be an expert here. If you were to think about cancer, what would you say would be the hallmark of cancer? Just cancer in general, no particular cancer. What would you say would be the hallmark of all of this? Okay, accumulation of mutations. What can somebody else add to that? What would you say would be the hallmark of cancer?
Cells that keep dividing.
Yes, cells that keep dividing. So unrestrained proliferation drives that, and as the proliferation occurs, then you get progression of cancer. You do have an increase in the number of mutations. All right, so we're going to try to address some of these questions in the next hour. Let's take first the normal cell. Our cells comprised of our bodies are comprised of all different kinds of cells, and I don't know whether you think about it, but these cells generally are turning over all the time. Some of them turn over very rapidly. For instance, the GI tract, very rapid turnover of cells. Those cells do not exist for that long, but that's in contrast to the brain, where neurons may last a lifetime. Your bone as an adult, maybe 20% of it will turn over. So there's a constant turnover of cells, and as you have the death of some cells, then they have to be replaced by others. But this is a very ordered response. You have a certain number of cells, and you will maintain that number over time. This contrasts with cancer, where you have unrestrained proliferation. So now we have both of the hallmarks that our very informed participants had mentioned. We have both unrestrained growth, so you have an accumulation of cells, and we have this accumulation of mutations that occur that make these cells even more dangerous than when they first were mutated. So to just try to get a few terms in order here, if we're talking about different types of cancer, we talk about carcinoma. Carcinomas are cancers that are derived from epithelial cells. These are usually cells that line a gland or an organ, or on the outside is your skin. So we have lung cancer, breast, colon, bladder, and prostate cancer, examples of carcinomas. And then we have cancers that occur in the bone marrow. We have cancers that involve the lymphatic system, and then some cancers, sarcomas, that involve fat, bone, and muscle. And you can be a little bit more specific with some of these and give them names that refer in particular to the type or where the cancer is located. So for instance, sarcomas are bone, lipid, muscle. So if you have an osteosarcoma, you have cancer in the bone. If you have a fat-derived cancer, it's liposarcoma. And then a couple of the carcinomas, adenocarcinoma and hepato being in the liver. So these are just some terms that you might see if you're reading about some of the literature that describes some of these cancers. Now you can see the development of cancer if you take a slide of a particular tissue, and you can see the change over time. And so a normal cell, or normal tissue, will have some cells that are restricted by a boundary, the basement membrane or a particular cell type. And those cells can proliferate. And this, we don't consider this to be dangerous. So for instance, if you get a callus on your finger, this is really hyperplasia or an increase in the number of cells that are there, but we don't consider that to be a problem. You can have proliferation, benign hyperplasia. Once the cells begin to look somewhat disordered, this is where you begin to show concern about what's happening. This is dysplasia. And then carcinoma in situ means that the cancer is restricted to a particular region. You see that there's this region down here. This can be a basement membrane, particular cell type. These cells are contained within the region where they're confined within a region. Then if those cells begin to migrate to cross this basement membrane, they become invasive, and that is a very worrisome type of cancer. So we're going to just take one example. We'll take the skin. It's an easy one. So you have this epidermis. You can see your skin, and you may not be aware, but you're constantly turning over. You're flaking off. We could find flakes of skin anywhere here. If you don't believe this, all you have to do is wear a cast for a couple of months, as I did last year. And then after they take the cast off, when I scratched my leg, under my fingernails were all the discarded cells. So it's a definite example of, you can see where the cells are being discarded, and new cells will come and move up, they'll migrate up this way and replace the old. So if you think about how cancer begins then in the skin, let's say we have a lesion here where there's a beginning of unrestrained proliferation. Here you can see that this basement membrane, that this layer of cells is being breached, and the cells are moving into the underlying tissue. This is where it becomes dangerous. And then once those cells begin to migrate into the adjacent tissue, they can then gain entry into the bloodstream, then come out at other locations in the body. And this is a particularly dangerous kind of occurrence. So if you have proliferating cells, you can have a benign tumor. This is nothing that we worry about. You just take that tumor out, and generally there's not any problem. But once you breach the region where the cells were normally located and they begin to traverse to other locations, you have a situation that can be quite deadly. So for instance, melanoma. Melanoma is a skin cancer. It's pretty deadly. You can have a tumor that develops on the skin, their darkened region, and that really isn't that much of a problem. If you can catch up quite early, you might be just fine. The problem is that these tend to migrate through the bloodstream to other regions. If they go to the brain, if they go to the liver, it is this kind of migration and metastasis that can result in death. So even though a person might die from the cancer that is present in the liver, it's not a hepatocarcinoma. It didn't originate in the liver. It originated here on the skin and traveled there, and it's a metastatic melanoma. So we want to, in order to try to recover, if you have cancer, what we want to try to do is detect cancer so that you have some chance of catching it early and alleviating any problems. Oh, and one thing we should mention is that a lot of times there won't be any symptoms of cancer. In particular, some cancers, ovarian cancer is one, pancreatic cancer is another. These are abdominal cancers, and there's not really much in the way of symptoms initially. It's only as the disease really progresses to a later stage that symptoms begin, and at that point, it is probably too late. So one of the
You refer to mutated cells. What is exactly a random mutated cell?
what is a mutated
What is
cell?
What is the definition?
If you can wait 10, 15 minutes, I'll show you exactly what it is, okay? Okay. All right, so we're going to talk about some of the general characteristics first. First is just an introduction, then detection, and then we'll get into how it occurs. And I'll talk about mutations, yeah.
So where is the cancer cell from?
Where is the cancer cell from?
Yeah.
Well, they can originate in many different places. If it originates, the example I just gave you, a melanoma originates in the skin. Hepatocarcinoma originates in the liver. You can have them originating in the pancreas. Most any tissue can have a cancer that starts there.
I mean the cancer cell has already is there and actually not be firing, or is it possible to divide it from our stem cell and got something in progress?
You mean what cell does it come from?
Yeah.
There are people who believe that there may be stem cells where cancer may originate. Yeah, but it can also be the resident cells and tissues where it originates. That's a question that is one of great interest to a number of people, and that will, in that one, you'll have to wait a few years to get the answer. Yeah.
I just heard on the radio this morning.
Okay, hot news.
Just heard on the radio this morning, and I don't know if it was the Olympic Channel. And, they said on the morning show that they did find that a lot of tumor growth is due to the stem, that tumors have their own set of stem cells that are differentiating, I guess, like it brings you.
Yeah, you will definitely find people who believe that, and then you will have people who have been in the area also who believe somewhat slightly differently. But
It was a news flash, so.
okay, well, we'll take that flash. That's our flash of breaking news. Okay, thank you for that. Okay, so I thought I mentioned one This occurs in women in the reproductive tract. So you can see here the reproductive tract, the ovaries, fallopian tube, the uterus, and at the base of the uterus is a cervix. And there's a cancer that can develop here, cervical cancer. And there was a Greek guy a number of years ago who figured out that if you just took some of the cells, used like a brush or a swab to get some of these cells from the cervix, and you looked at those under the microscope, you could tell whether cancer was present just by looking at the cells. And this very simple method resulted in a 75% decrease in the death rate. So this is a very substantial change. And just to give you a little better look at what you might see if you looked under the microscope, these are cells from a person with a normal cervix. So you can see the cells similar in size. Here we have an abnormal pap smear. Notice in particular the differences in the sizes, and we'll talk a little bit more about this as we go on. So this was a very important advance, one of the first advances to try to screen for cancer. And then if there is any question about whether to get an abnormal pap smear, another test that is used is one in which you look for the presence of the human papillomavirus. This is a virus that infects these cells, and the presence of this virus is associated with an increase in cervical cancer. So that's another marker that cancer might be present. All right, so we're going to talk about the big three. Biggest killer is lung cancer. And if you look just with a simple x-ray, you can see if there is a tumor there. If you want to get a better picture, you can get a CT scan where you take a series of x-rays and you can get the 3 dimensions of what it looks like. So these screening methods are used to look for lung cancer. The next biggest killer in women is breast cancer. If you are screening there, you're using a mammogram. This is essentially an x-ray that you compress the breast and you use the x-ray to take a picture so that you can see if there's a tumor present. In the male, if we look at the male reproductive tract, you can see the bladder here. This is the urethra that leads out, and the prostate gland is located right here. This is the second biggest killer in males. It's very common as a male ages that the prostate gland will increase in size. That's common. That's called benign prostatic hyperplasia. And this is something last year when I gave this lecture, I would say that the way to screen for this is to look for a protein that's produced by the prostate, and that's prostate-specific antigen. But this is under a great deal of controversy at the moment as well, because they have used the PSA test. It's just a simple blood test. Then you look for this secreted protein, prostate-specific antigen, and as the prostate begins to grow in size, so if you have benign hyperplasia, you can have a rise in PSA levels, and then cancer of the prostate is associated with increased levels, even more increased levels of PSA. But there is a great deal of controversy now whether these should be done, because it's thought that a number of men have had prostate cancer surgery, which can lead to incontinence, and it's thought that maybe it is better now to just try to wait. It's called watchful waiting, to see whether the tumor increases in size. You can do a rectal exam to figure out what the size of the prostate is. But this is, again, something in science, there's not always black and white. We're in a gray area here as far as PSA levels are concerned.
You said that prostate cancer is a hormone-driven cancer.
Prostate, yes.
So I don't, this is something that's always puzzled me. I thought men's levels of testosterone decrease as they age. So what hormone exactly advances prostate cancer?
Okay, so we're going to get into some hardcore endocrinology here for just a second. Men produce testosterone. That's what is involved in increasing muscle mass and red blood cell count, and then testosterone can be converted. It is converted to another hormone, which is called dihydrotestosterone, and this is what targets the prostate gland. And in fact, I don't know how, you stop me if I get too much into this, but if PSA levels rise, they will rise under the influence of dihydrotestosterone. But testosterone gets converted to dihydrotestosterone. So there is a drug that can be used that will decrease the conversion, inhibit the conversion of testosterone to dihydrotestosterone, and then the levels of PSA can go down. And in addition to that, the dihydrotestosterone is responsible for male pattern hair distribution. So these guys can get some of their hair back, which they like also. So that's we have a benefit there.
I saw you looking at us.
No. You are too good looking man. Okay, so this is a test which is being done, and the hope is that we would be able to have more specific tests to determine whether it's a cancer that's going to progress and be very aggressive, or one that can be left alone. All right, the other of the big three is colon cancer, and since I thought maybe not everybody knows exactly what their digestive tract looks like, you have food that comes in, goes down the esophagus, into the stomach, the small intestine, hundreds of feet of intestine, then through the colon, we've got the ascending, transverse, descending, and then sigmoid colon. And in order to try to screen to see whether colon cancer is present, you could do a couple of tests. One is a simple at-home test. The doctor will give you one of these kits here. You take feces, you put it on this little strip here, put it in an envelope and send it away to the lab, and there they look to see whether there's blood present. There should not be blood in the feces. If there is, that's called the fecal occult blood test, so that's an easy one to do, even screen lots of people, doesn't take too much time or money. But if you detect some problem there, then you want to go further and see what is occurring, and with that you can use a colonoscopy in which you insert this laser, what do you call this, laser fiber optic, thank you, I couldn't get the word, fiber optic device, and you can actually look and see whether there's something that's present there. These are some examples of colon cancers here, and as these progress, they become more angiogenic. They need the blood supply to feed the tumor, and you can see the presence of these blood cells. So there are a couple of ways that you can try to look early, find a polyp before it develops into a cancer. So there are a number of tests, and you use, for instance, with the pathology, you can look for cervical cancer. If you think that it might be cervical cancer and you want to do a DNA test, you can look for the presence of the viral gene from papillomavirus. There are also proteins that you can look for. You can look for PSA in the blood. So you can use a number of different methods using blood or tissue to look at DNA, proteins, and then basic histology or pathology to see if you can find the evidence that cancer might be there. So now I want to go back and try to show you what you might see under a microscope, and I'm going to bring back the pictures of the cervical cancer. Yeah?
Yeah, excuse me. In the last slide, you mentioned about lots of of the way to detect the cancer. But what's the point for we to go further? What's the improvement can we make between this? For example, if I look for the pathogen, what's the defect of looking for pathogen?
Why would you want to look for the
No,
pattern?
no. I mean, once it's not enough, we recently do. Yeah, because you introduce lots of way to detect the cancer. Although we can detect those cancer now, but what's the improvement we have to make?
Well, the improvement in far as prevention or detection?
No,
What?
detection.
Detection, oh.
Yeah, though we have lots
Oh.
of way to.
Yeah, these are the ways that we have now.
Yeah.
And certainly,
What's the
you can
problem
use
with this?
nano devices to look in blood for very small levels, what?
No. I
No.
mean, what's the problem with this detection technique? No.
Well, the problem with the detection technique
Yeah.
is they're not sensitive enough.
Oh,
Yeah, I mean, if we could get greater sensitivity, if we could identify them earlier on, then the chances of cure would be greater.
but the genomic way can detect these.
Oh, okay. So, for instance, you can have a genetic disorder, and let's say breast cancer is one that I'll give as an example. This is handed down from generation to generation. If you look at the family tree, there are people in every generation that have breast or ovarian cancer. And if you know that you have that mutation, you can test now for that mutation, women will have their breasts prophylactically removed because there's an 85% chance that you'll get breast cancer if you have this mutation. So that kind of thing, if we could find markers for genetic disorders, that kind of thing can be done. But we don't know what all the possible mutations are that can occur. Yeah.
Just follow the question is that, Jenna, I know the problem is infection for cancer, and how do you think which way is the most sensitive to infection? Like initial product, the skin, which way is more sensitive to cure for cancer?
Well, the most, you're asking what is the most sensitive way?
Yes, for early detection. Yeah, I know that there are a lot of methods.
Yeah, it really depends upon the cancer. I mean, some of the cancers, like lung cancer, is another one where you might not have symptoms, and so we don't screen everybody. Not everybody gets an X-ray of the lung. There's not large scale. The most sensitive would be dependent upon what kind of cancer you're talking about. If you have this mutation I talked about for breast cancer, you can very systematically, very sensitive, determine whether there's a mutation in that one gene. That's extraordinarily sensitive. But the problem is that accounts for a very small portion of the breast cancer that exists.
Is there any indication for cancers?
Is there a prediction whether, that's something that's being worked on now. He's asking whether we would know whether a tumor is going to metastasize. That's exactly something that they'd like to do for prostate cancer, to figure out, is this a cancer that's going to be kind of sitting around and just growing a little bit, or is this going to be something that's going to go to the brain and kill this guy? So what they're doing now is trying to use personalized medicine to look at the gene expression profile of tumors and to predict from the profile of genes that are expressed in the tumor cell to see whether or not that is going to be a particularly aggressive cancer. But at this point, that's a, something that's in the development and not something that is carefully defined, which we hope it will be in the future. Yeah.
I have a two-part question.
Yeah.
So part one is, when was this breast cancer gene discovered that you're talking about?
I'm sorry.
Or, oh, it's okay. When was this gene that you're talking about for breast cancer discovered? So like when was the starting point that women had the option to proactively remove their breast tissue?
Let's see. I actually have, I have a slide of that in a little bit. There was a woman, Mary Claire King, who worked on this for years and years. And she knew that it occurred on one chromosome. and she narrowed down the region over a period of decades to figure out where it was, and then it was actually a group, another group that identified using this population of people that was more restricted. They could figure out that it was this one gene, breast cancer 1 susceptibility gene, and it was it 70? Do you know? 90s? Well, she started her work in,
Salt Lake City period.
right. Okay.
So it
Late
might,
90s.
yeah. It might be too early for part 2 of my question, but I was just wondering because something that's always been puzzling me is, I tend to think of the human body as a resilient, being, and I guess I always just wondered, even though technically I know that this gene exists, why someone would voluntarily remove their breast tissue, because, you are removing glands, so I guess in certain respects you're removing potential cancer sites to grow, but if you still have the genes, then unless you take out every single last breast cell, is that really a guarantee that you're not going to get breast cancer? It's only 10, 15 years ago that they started doing this, but.
Yeah, I mentioned that breast cancer is a hormone-driven cancer.
Yeah.
And so there's a less humongous way of dealing with this. If you have the BRCA1 mutation, then you can take, since estrogen drives the proliferation of cells, you can use an anti-estrogen to restrain that a little bit. And that, if you give a woman the anti-estrogen tamoxifen, those women who are at increased risk will have a 50% reduction in breast cancer. So you can do something less than that. I mentioned that as the most.
It was just something that I forgot that I was always like, how would they do this?
Yeah, the human body is extraordinarily resilient, and we probably get cancer many times over, but we have mechanisms, in particular DNA repair proteins, that are involved in constantly getting our system back so that we don't have that problem. Yeah.
So I have 2 questions. One, what is the breast tissue just constitute the fat of lymph nodes?
No, they're in a little bit further on. I keep saying this, don't I? A little bit further on, I have a picture of the composition of the breast tissue, and I'll show you exactly where that comes from.
Okay, because like guys who get breast cancer, trying to figure out what portion.
Usually if a guy, I don't know if it's usually, but a male who gets breast cancer may be involved in this, may have in his family tree BRCA1 mutation.
And then the other question is, our genes are constantly trying to fix it. Why doesn't it mutate back if it's
And the mutation is generally a random event. So, for instance, if you're exposed to something, it can cause some mutation. But then we have in our cells proteins that can recognize that maybe the DNA molecules are not paired correctly. So an A will always go with a T, a G will always go with a C. And if that's not paired correctly, they'll go in there and they'll fix that. Or if they see that there's a nick, they will fix that nick. If they see that a base has been removed, that there are many different DNA repair mechanisms to try to maintain the integrity of the genome. Okay, so if we look at just these, the cells that we get from the cervix, you can see a number of characteristics that are present in normal cells and are present in the cancer cells. So you can see a change in the sizes, a great variation in the sizes of the cells. The nuclei are similarly sized here, but you can see variations in the size here. The cytoplasm accounts for a great portion of the cells in the normal cells, but that can be greatly retracted. So you have a tremendous variation in size, shape, cytoplasmic composition, and the nuclear, the appearance of the nucleus. There can be, for instance, in brain cancer, you can lose some of the specialized features of the cells. And then in cancer, there's generally a disorganized arrangement and poorly defined boundaries. And I'm going to show you an example of that using the breast. Here you go. So this is, the breast is an organ that is very functional during the time of lactation. This is after pregnancy, delivery of the baby. Then this gland begins to lactate. And this just shows these lobules that comprise the lactation apparatus. And inside each one of these lobules are alveoli. And this is a single alveolus. And you'll see there, so it's kind of like you have this grape-like cluster. And if you look at one of the grapes, you have that lined by a single layer of epithelial cells. Epithelial cells line the interior. And so these produce the milk. Outside of that are myoepithelial cells, myomuscle. They are going to cause contraction of this alveolus so that the milk is ejected into the duct and can be expressed. So this is the functional unit. Now let's look at, if we have already considered the alveoli which reside in here, let's look at the duct. The duct is arranged in the same way. We have a single epithelial cell layer. As a woman ages, though, sometimes you'll get some benign hyperplasia, so you have an increase in the number of cells, not just a single layer. And then if we move down to ductal carcinoma in situ, you get a proliferation so that entire duct can be infiltrated or can be populated by more epithelial cells. And then as the barrier begins to break down here, you get micrometastasis and then full-blown metastasis to other portions of the body. So we're going to look at 4 different kinds of stages of going from normal to the invasive breast cancer. We're going to look at normal cells. These are tissues which we obtained from mammary reduction surgeries. This is benign hyperplasia, so a little bit of proliferation, ductal carcinoma in situ, and invasive breast cancer. And these are some tissues that we were staining. This is the brown indicates that these cells are proliferating. This is expression of a protein, proliferating cell nuclear nitrogen, PCNA. And you can see here, look, here's a nice duct. You can see a single layer where you have the epithelial layer surrounding the duct. If you look here, we're into benign hyperplasia. We have multiple layers of cells now, but still not considered to be problematic, but this is where you would have watchful waiting. Ductal carcinoma in situ, you can see this is almost completely populated by these epithelial cells, but you can see that you still have the integrity of the outside membrane, and this is invasive breast cancer where you see really a disorganization that is occurring. And you can see the variation in the size of the nuclei here, which is a characteristic of cancer. Now this is another one where you have, did you have a question?
Oh, I was just wondering, do you know what the typical turnover time for those vesicular epithelial cells is in the tissue? I know
Of a normal
Yeah.
epithelial, breast epithelial. I don't know. We work with breast cancer cells, and they have a 2.5-day turnover. Do you know the turnover of a normal? You know, we can't really, normal cells don't grow very well in culture. They're much more difficult to figure out what the half-lives of those are. I would expect that they would be longer than 2.5 days, though. Okay, so this just shows you another protein. This is actually a protein that is involved in, it's a chaperone. It helps maintain protein structure. And you can see that it is very well organized around this alveolus. It's not in the epithelial cell layer, though. It's on that myoepithelial cell layer that surrounds it. And here you can see you still, even in the benign hyperplasia, you still got appropriate localization of that. But once you get into ductal carcinoma and invasive breast cancer, you no longer have this very strict localization. So we have a number of different characteristics. Just by looking at a microscope, a lot of times you can tell whether or not you're dealing with a normal cell or a cancer cell. So if you think about some of the causes of cancer now, we have intrinsic factors. I changed the order of these a little bit just because I wanted to talk in a specific order. So one is hereditary, which we already mentioned. You can get a gene from your mother or your father that is going to increase your incidence of getting breast cancer, the risk of getting breast cancer. As we age is another consideration. Hormones, both in prostate cancer and breast cancer, and our diet is important. These are all some intrinsic factors. And then there are chemicals that we can be exposed to, viruses, bacteria, and radiation. Yeah?
I wasn't sure, I'm not experienced, so I don't know if this is kind of obvious, but when they say, when you say hereditary for factor of getting cancer, I know they're usually caused by mutations, so what is it usually that's passed down? Is it that a body doesn't have enough of a check to eliminate mutations? What is usually the passing down of the gene that could cause extra risk?
You're right that few cancers are caused by heredity, but there are some that we know of, and one is the breast cancer gene. Most of them are mutations, and in particular, which I'll talk about in a minute, mutations in particular proteins that are involved in restraining growth are very much involved in cancer progression, oncogenesis, and cancer progression. So the mutations, most of the mutations are not passed down, but some are. And those that are more random or that affect other proteins are generally caused by some of these other factors, chemicals, viruses, and radiation. Okay, this is a quiz. What one thing could you do that would dramatically reduce cancer, reduce cancer? And I'm thinking of one specific cancer, maybe that's a clue. Not smoke. That's exactly right, the biggest killer. Because, and why is that? If you look, because tobacco. Does anybody in here smoke? Don't smoke around me.
Not anymore.
As of now? Okay. So the reason that smoking causes cancer is look at the number of chemicals that are in tobacco smoke. These are carcinogens. So smoking is the main cause of lung cancer. It contributes to other types of cancer as well. It's responsible for 1 in every 3 cancer deaths, and it's the largest single cause of death from cancer overall. So don't smoke. All right, if you look, sometimes it's not really apparent though that this is the problem because there's a lag time between the time you smoke and the time you get the disease. And that lag time can be a little bit, maybe it can make you comforted a little bit. While I'm smoking, I'm not getting cancer, but wait for 20 years and the incidence of cancer rises as the number of cigarettes that you have smoked rises. There's a relationship between the number of cigarettes and also the incidence. Another thing that we really can't do anything about unless you die early is you age, and age is one of the primary risk factors for cancer. If you look, here we have just 2 examples of an increase in breast cancer with age as you go to 80 years old, increase in colon cancer, and if you take a 75-year-old, compare him to a 25-year-old, that 75-year-old has 100 times the risk of getting colon cancer of the 25-year-old. And that's because of age. You might get a better idea of this if you look just for some breast cancer statistics. If you're 25, 1 in every 20,000 women might get cancer. Go a decade, it's down to 1 in 600. Another decade, less than 100. If we go by decades, 33. And so your incidence of ever getting cancer as a woman is 1 in 8. So if we count here the number of women, we could calculate about how many of us likely get cancer before we die. Radiation from the sun is a risk of getting cancer, mainly melanoma. Dallas, which is very sunny, has a higher risk of getting skin cancer than Detroit, where it's cloudier. If you look at radiation, this is high energy radiation. Those people who went into the Fukushima plant to take care of getting that reactor shut down, their incidence, their risk of getting getting cancer is much higher than residents who lived in that area or people who lived in the southern portion of the country. So an interesting thing here, I think, in particular for our visitors from around the world, is that different parts of the world have different risks of getting different types of cancer. And so if you look in China, Taiwan, liver cancer is a big problem. Brazil, cervical cancer. Australia, sunny all the time. Skin cancer is very high. Lung cancer in the United Kingdom. And then we're just going to take two comparisons here. Japan, where stomach cancer is quite high, and the US, where colon cancer is quite high. And there's a very interesting observation that has been made. Japanese have a very low risk of getting colon cancer. People in the US have a higher risk. But if you take somebody from Japan and you move them here, that Japanese person is going to assume a risk almost as high as those who live here. If you take a person from Japan who has a high incidence of stomach cancer, the US, the incidence is quite low. You take that Japanese person, put them here in the US, and their risk declines. So this is one argument that is certainly not all heredity, but there are other things involved which could be cultural, behavioral, or environmental. So there are other things that which we don't understand exactly why those occur. One way that cancer can occur is through viral infection. So you have viruses that have RNA or DNA. The DNA can be integrated into the human genome, and if that infects it, it can cause changes in genes that regulate cell growth and initiate cancer. One that I mentioned already is the human papillomavirus. This is what has been linked to cervical cancer. And the human papillomaviruses pass through sexual contact, and the infection of the virus increases the risk of cervical cancer. Hepatitis is linked to increased rates of liver cancer. Kaposi sarcoma, I think, is probably something that you've heard about, and there may be some misunderstanding here. It's associated with HIV/AIDS. So if you have someone who's not infected with the human immunodeficiency virus, they don't have Kaposi sarcoma. But once infected with the HIV, they have a depressed immune system, and now if that virus infects, they can get a cancer of cells that line the blood vessels, and they get these skin lesions, which look like this. And as this disease progresses, can ravage the body. In addition to viruses causing cancer, you can have bacteria that can be linked to an increased risk of cancer. And this bacteria here is called helicobacter pylori. It was first found to be associated with an increase in ulcers, but there's also an increase in stomach cancer. This is the stomach. Sometimes the duodenum also can get, you can have an ulcer there as well. You can see here, there's an ulcer here that's been formed in the stomach, and this is a cancer. And the incidence of stomach cancer increases with infection of helicobacter pylori. Okay, now somebody asked about DNA sequence and what does that mean or how does a mutation take place? So in every cell in your body, you have 3 billion base pairs. This is the template that is used to produce RNA. The RNA is used to produce protein. If you mess up the template, if you mutate the DNA, then the RNA is going to be changed and the protein is going to be changed. The proteins are the functional units in the cell. They are what make things happen. They are what keeps that cell intact and keeps it regulated in a normal way. So we want to maintain the integrity of this sequence, which is comprised just of 4 different bases, A, C, G, T. If you have a mutation, you can have just a single base change. And sometimes this isn't a problem because it won't actually change. It'll change the RNA sequence, but it doesn't change the protein. I have to go into more detail, but for now we'll leave it at that. A bigger problem comes if you have some bases inserted or if you have some bases that are deleted. This can take a protein and really change its character. So now we're going to talk about this breast cancer associated gene, BRCA1, BRCA2. So this is a gene that's hereditarily, is that right? Yeah, carried on, and it's not responsible for the majority of breast cancers, but it is certainly responsible for some. And this is in a very crude way, if you just took the amino acid sequence of this protein and laid it out here, this is the sequence, this is the amino acid sequence of this protein. Oh no, that's a gene sequence, excuse me. And we know that there are mutations that can occur in this protein, and these all indicate places where mutations have been found. But it's very interesting that in descendants of the population of Eastern European Jewish people, those women have a higher incidence of deleting 2 nucleotides, an A and a G. This doesn't seem like that big of a deal. If you have a huge protein, what's the big deal if you get rid of 2 proteins? Well, or 2 amino acids. It is a big deal, 2 nucleotides. It is a big deal because it causes a frame switch. And now, rather than producing a protein that's this, or that would account for this big, let's say this big, because that's the gene, now you're producing a protein that's only this big. This protein is involved in DNA repair. This isn't. So you have a function that is lost in that cell. And if you look, this is a family where the BRCA1 mutation was present, passed down to this generation of girls. Here you have 4 beautiful little girls. In 1986, she was diagnosed with breast cancer. 1991, diagnosed with breast cancer. 1992, diagnosed with breast cancer. You can see this pattern where the number of people, not all children are affected, but it can really ravage a family. And it occurs not just in one generation, but because it is passed down, whatever it is passed down, it can go from one generation to the next. Yeah.
Do you have any, so in the same population, we have a higher incidence of T-cells, and the advantage of being a carrier for the disease is that your chance of getting tuberculosis is low. So during the whole last time, we were looking at those that carry this mutation, we have tuberculosis. And do we know if, since we're in the population, is there any benefits?
Yeah. Well, you can have some side effect like that.
We're probably on to repeat the question.
Yeah, she's saying that the Ashkenazi Jewish population, the descendants, they have a mutation that causes Tay-Sachs disease. But those individuals during the Holocaust didn't get tuberculosis like the general population. So you actually have some protection even though you have a mutation. Actually, the Ashkenazi Jewish population has several several of these anomalies that have been passed down from generation to generation. And the thing of it is, you had a population that was reproducing within a very restricted group. So any mutation would be passed down again and again and again. Whereas if we marry outside of a population, then a lot of times you'll get at least one gene that's functional, one that's not. So it's an advertisement for breeding outside of a very restricted population. Not that I'm, I don't want to influence you in that way. But okay, so if you talk about genes in cancer, you have these hereditary associations that can occur, but chemicals, virus, radiation can also change the DNA sequence. And in particular, there are some genes that are very much involved in the development of cancer. In a normal cell, you have the normal complement of genes, and those cells are going to divide on a regular basis. If you have a cancer cell and you get a mutation of DNA of a proto-oncogene, you have an oncogene then that can accelerate cell growth and division. So let's think about why that would happen. If you have a normal cell, you have, let's say this is a cell, here's the outside along there, and in the yellow jacket, you're the nucleus, so stand up. Okay, so I have a message. I'm a growth factor, and I am going to be involved in getting growth of this cell going. So I'm going to say, I'm going to send you a signal. I'm a growth factor, and I'm going to bind to a receptor, and that receptor is going to give the signal. Yes, this is exactly right. This is the way it happens. You have a signal that comes from the membrane and goes through multiple partners to get to the nucleus to turn on gene expression. Perfect. Okay, so there are going to be even more partners that are involved here. So you have, here's the receptor on the outside of the cell. The growth factor binds to the receptor, and there are multiple intermediary steps here to get to the nucleus that then involve turning on gene expression. And so what happens if you have a proto-oncogene, which is the normal gene, being converted to an oncogene, a mutation in that gene. So what that's going to result in is a protein that looks like this, turning into a protein that's different looking. Its function is going to be different. So now, if you look at what happens in the cell, you can have the receptor that is really not too functional, but a very active intermediate. Any one of these intermediates could be constantly sending signals to the nucleus to increase proliferation of the cells, and that then drives the proliferation. And even though that receptor is no longer active, some of these intermediary steps, these mutated proteins are going to be turning on gene expression. So that's what happens when these proteins responsible for fueling growth change. And these oncogenes accelerate cell growth. But we also have in our cells proteins involved in inhibiting growth. So you can have proteins that inhibit the receptor, some of the intermediate proteins, so we grab onto them, keep them from activating, and they tend to limit the growth of the cells. You can have a situation where now you have these proteins that might normally try to restrain the intermediaries or the receptor, and they become mutated. They're no longer able to restrain that. So now again, you have increased cell growth because the tumor suppressors are no longer functional yet.
Why is it going to take so long for cancer to develop in case of expressing intergene if they have the damaged DNA repair? Why would it take 4 years? Why would it be much sooner?
The breast goes goes through a very big remodeling process during adolescence, during pregnancy, and then during lactation. And it, you know, the very interesting thing about it, the reason that Mary Clark King was able to figure out that there was a breast cancer associated gene was that she took the women who were 40 or under and looked at their family trees. And they actually, those women who were 40 and under got, they were the ones generally who were more generally had the BRCA1 mutation. If you look at the older population where you get the effects of aging, you don't see that as much, but in fact, in the younger women, the BRCA1 mutation did increase their risk of breast cancer. It does take a while to develop. All right, so one tumor suppressor gene, which you may have heard about in one of your classes, is P53. P53 is really important because it will cause a cell to apoptose, to die. And that's what you want to happen if you have a cell that's going on accelerating growth. So I mentioned DNA repair proteins. These are really critical proteins involved in keeping the cell template, the DNA, making sure that it is exactly pristine and the right sequence. Sometimes you can have mismatches where a T is no longer associated with an A. That's wrong. You'll have a protein that will recognize that. Or if you have a base missing, or if you have insertion or deletion, those kinds of changes can occur. This shows the localization of one DNA repair protein in some breast cancer, invasive breast cancer. The thing that's troubling about this is that if you look here, you really don't see any brown. You really don't see the presence of that DNA repair protein. And that tells you that this protein is not going to be involved in helping to repair DNA, and thus you will tend to accumulate more mutations. So we have a situation here where we have a normal cell. We want to maintain the integrity of the template. So we have DNA repair proteins that repair it, that brings it back to its normal state. If you don't get repair, maybe the mutation will cause something to occur, so you get apoptosis, the cell will die. So again, you don't have cancer. The problem ensues when you have mutations that occur, mutation upon mutation. You can get increased uncontrolled proliferation and then invasion into other tissues. Yeah.
Is there any other response of the body to mutations besides the repair?
That's the primary response, is because there are, in order to get a response to mutation, you have to have a protein that's in the nucleus. And there are particular proteins that are located in the nucleus. It's very important for a DNA repair protein to be in the nucleus so that it can survey the template and see whether there are mutations. And that has to occur there. It's mainly, that is mainly it. That's our defense system.
If we have all this increased proliferation, wouldn't the cells be fighting for nutrition?
Yes.
Cause cell death?
If we have increased proliferation, will the cells be fighting for nutrition? Exactly right. And so what is one of the characteristics of a tumor? So let's say you have a melanoma, it goes somewhere else, begins to form a tumor, and it is starved for nutrition. So what happens? Angiogenesis. So you will see the angiogenesis, blood vessels have to get there because oxygen can only diffuse a particular distance, and you need nutrients, and so you have to get you will get increased angiogenesis, increased formation of blood vessels into the tumor.
Are there good situations where that happens, that increased blood flow, or like really high concentrations of increased blood vessel in a certain area, or is it only with tumors that really happens?
Let me think about that. Yeah. Oh, yeah, sure. If you have a wound, wound healing, yeah.
oncogenesis, or is that potentially an area where the tumor would have its own stem cells to generate its own vascular system?
No,
Or
this is actually a
stealing from the.
No, this is actually a system of chemical signals that are sent. So in those cells that are deprived, they will send out a message, vascular endothelial growth factor, VEGF. That then causes those blood vessels to migrate and to come to the tumor. So it's really extraordinary that they can send the signal, the blood vessels can respond and come that direction.
So if the person is unwounded, could cancer be diagnosed by measuring the amount of VEGF, is that it, in the system? Because if it's calling for all these extra nutrients?
Yeah, I don't know that the peripheral levels of VEGF would be increased. You know, a lot of times it's just a little distance. I mean, the capillaries are most everywhere because oxygen can only diffuse a short distance. So the capillaries are going to be real close. That message only needs to go a little distance to get those blood vessels there. It's not going to go through the blood generally. Yeah, it's not, I don't think you're going to measure differences systemically. Yeah.
Yeah, I think she's saying the right thing because during the brain cancer, sometimes we use MRI to diagnose that because the angiogenesis there will be very affected, and those vessels are going to be a little leaky because the wrong message to let them go growth. So the MRI can be an early detection of those brain cancer.
So the MRI, are you saying? What are you saying?
MRI.
Oh, MRI, yeah. He said it could be used for detection, early detection of brain cancer. What time are we supposed to stop here? Now? Okay. You know, okay, tobacco, don't smoke. Don't drink and smoke.
We have about 15 minutes discussion, so.
Okay. Would you like, should I just end it there? Should I go through quickly this last section? Sure, which? Go through? Okay, I'm going to do this. So we have these DNA repair proteins that are instrumental in maintaining the DNA template, in particular, maintaining the integrity of tumor suppressor genes and fertile oncogenes, so we don't get the oncogenes to drive proliferation. And then we want to talk just a little bit about how we can prevent cancer. One is to limit tobacco consumption. I think you got that. The second, there's really this synergistic kind of thing between alcohol and tobacco, though. So if you drink 4 drinks a day, or if you consume 2 packs plus a day, then you have an increase in esophageal cancer. That's the tube that runs from your mouth to your stomach. And if you are both a drinker and a smoker, you get a 40-fold increase in the incidence of esophageal cancer. Okay, if you look at the incidence of colon cancer, it's really quite high here in the U.S. We said before, we're one of the biggest consumers of meat. And if you look at the meat consumption, there's a really pretty good correspondence between the amount of meat that you consume and the incidence of colon cancer. So Japan, which had a very low incidence of colon cancer, they consume significantly less. And certainly it's important to protect yourself from the sun, but I actually get a little sun. I synthesize my own vitamin D. So that you want to make sure you don't have excessive sunlight so that you are exposed to increased chances of getting melanoma in particular. There are basal cell carcinoma, squamous cell carcinoma. These are cancers which are not as problematic. And avoiding viruses, so for instance, human papillomavirus, which is passed through sexual transmission. Limiting the number of sexual partners can help to decrease the incidence of the virus of cancer, and certainly in areas where you have increased liver cancer, that's also associated with increased infection with hepatitis, so you can try to limit your exposure. There are a number of chemicals which you try to make sure you don't come in contact with. This is mainly due to occupational exposure. If you are in some of these occupations where you're exposed to these chemicals, the incidence of a varied number of cancers can increase. And then even if you aren't working directly in the industry, if you're living outside of it and exposed to those toxicants, you can have an increase in cancer. So if we look, what we want to try to do is maintain the integrity of the DNA template so that the cells are normal. If they're not, we can rely on DNA repair proteins. We can rely on tumor suppressor proteins to help us out of this situation, either through DNA repair or through apoptosis, and that will help to maintain normal cell population. I'll stop.
We do have some time for discussion.
Yeah.
So I've heard that cancer cells usually handle multiple copies of the chromosome instead of just the regular two copies.
Yeah.
So and you mentioned as well that the cancer cells typically have a morphology where the nuclei is abnormally enlarged and the cytoplasm.
Copies of genes in
chromosomes.
of chromosome, even
copies of
as the mutations accumulate, you can get really kind of extraordinary aberrations in the cancer cells. Where you can have some parts of the chromosomes that are duplicated. You don't have the normal number anymore. It's not like you can do a spread and look at the normal chromosomal population. And further along you go, the more aberrations you can pick up.
I was just wondering, so let's say if you wanted to do genetics of cancer cells, for example. I mean, with such multiple copies, what's the, the enhanced issues that you might face?
Okay, so now you're talking about increased copies of some proteins?
I mean, not
Or
proteins, but
of some DNA regions?
like this chromosomes of DNA.
Yeah, it doesn't have to be the whole chromosome. It can be part of it or a region. For instance, there's one type of breast cancer where you have the amplification of a particular gene called HER2/neu. And if you have that, it actually is one of these receptors that is involved in maintaining growth. So instead of just having one of these receptors here, you're going to have lots of them. And that really fuels growth. The good thing about that is that you can use an antibody and you can restrain the activity of that receptor so that it doesn't fuel growth. There are many examples of aberrations, especially as the tumor progresses, and there has been, there's this initiative to sequence the genomic DNA of normal and tumor DNA of individuals who have different types of cancer. So what have they done now? Pancreas, brain, and ovary. Yeah. So what they did was sequence a number of these, and then if you can line those up and see, are there common features of these, and what are the features, and is that something you could target in some way, or even detect in some way? Yeah. What can we do for cancer therapy in the future?
Nanotechnology.
Oh, nanotechnology. Oh, there you go. We have an expert who's going to tell us that in just a little bit. I think I think your imaginations is the limit to what we can do. Really, if you know the technology, you dream about what could I do? What could I do to detect it, or what could I do to
For
treat it?
fail, for cancer fail.
For a cancer cell? Yeah, I mean, well, cancer therapy. Okay, so one of the real problems is that a lot of so if we got the cell again, a lot of the proteins are inside of the cell, which is a problem for us because, they're hard to get to. One of the things that nanotechnology can do if you have little particles is to ferry something into the cell. Now, if you have, let's just say you have a particle and it has something that recognizes the receptor that's on a particular cell, and you want to target that cell, well, you attach to that something that will get inside the cell. You can kill it off if it's a chemotherapeutic agent, or if you want to try to regulate your activity, I could go into the cell, target it here, get into the cell, and target a particular protein that was involved in some cascade that regulates cell growth or metastasis.
It's really a problem for targeting. So,
Targeting is a huge problem. Yeah.
Is there any way that can be fruitful and particle?
Yeah, targeting is, I think that's the biggest thing. Because you would have to find something that's present only on that cell, that cancer cell. If we could do that, then we could do lots of things. And it's probably going to be different for different kinds of cancer. Yeah.
Do we know of any other biomarkers besides the HER2 for breast cancer that, like receptors?
Oh, sure. The estrogen receptor, the progesterone receptor. The estrogen receptor is targeted with the anti-estrogen. That's a real nice one because it doesn't have that many side effects. It only targets tissues that have the estrogen receptor. So it has specificity involved in it. But it will limit disease recurrence by 50%. It decreases the incidence in women who are at high risk by 50%. So it's effective, but there's 50% are effective. That leaves another 50%. So that's what you guys have to work on.
Just changing topic a little bit, but I'm curious, cancer cells, something that obviously something to do with their structure, but why, and their mechanics, why are they so mutated? What is it usually called? Is it a mutation that causes something quite differently in its structure, for its environment?
Yeah. So why do they look so different?
Yeah.
There are so many things that are so different in a cancer cell, and there's an architect, there's kind of like a skeleton that keeps the cell looking normal, and that is one of the things probably that's just no longer well regulated, and so you get this change in the morphology of the cell. Oh, we'll take one more. Yeah.
So I have said that most of the cancer deaths is caused by the metastasis, right? But I'm not sure.
Deaths caused by metastasis, yeah.
But I wonder, what are the details of, what happens, for example, when you have metastasis in different parts of your body, how, it will, these cells are not gonna, produce some of the chemicals or growth factors in the body, or what is the main cause of death when you have metastasis? If you stop producing something, or you overproduce, or is it just a mixture of everything?
Yeah, once he's asking about metastasis, why, when it goes to different parts of the body, what causes death, or?
No, I understand how it happens, but I don't understand exactly what is the cause of death of a person. It depends on the organ, right? For example, if you have cancer in your bone, then you metastasize in other parts. But then, the function of this organ will be affected, but how much is that effect that it will cause a person to die?
Yeah. Well, if you get metastasis in the liver, for instance, this is an organ that can be very severely compromised. You have a lot of enzymatic function a lot of enzymes that are really valuable to maintaining your normal metabolic state, and that can be very deadly. If you have metastasis to the brain, it's very problematic because you've got a skull that encases all the brain, and if you have a tumor that begins to grow, it's going to press on something and alter the, compromise the function of particular brain regions. And, it's this, partly the, just your skull and the constraint of the tumor, and it's, it can cause changes in vision because the optic nerve is affected. If you have a prolactin or something like that, you can have different brain regions that are affected because of the tumor impinging on their function.
If we can control the angiogenesis, then we can hopefully control the cell metastasis. Is that correct?
Not exactly. Okay, if you can control angiogenesis, you can control metastasis. They're kind of different things. Metastasis is going to occur. You don't. Well, so maybe what you're thinking of is this, the blood vessels coming into the tumor and then allowing it to get out. Yeah. Yeah, there is a drug on the market now that they've used. It's effective for colon cancer and limiting angiogenesis, and it had been used for breast cancer. Thinking that if you can decrease angiogenesis, that would be useful, but it hasn't been shown to be any not effective. So either it's not effectively inhibiting angiogenesis, or it's not effectively inhibiting. I don't know what just went on my brain there. It's gone. We'll leave it there. Yeah.
There's one approach, which is to cut the blood supply, essentially the supply of the nutrient in order to starve to death or to limit against growth. I think there is one particular mechanism that people are targeting.
Yeah.
Cancer treatment.
The problem with that, though, is if you have a tumor and you're going to cut off the blood supply to it, things may happen between the time you cut off the blood supply and some cells could leave that you would probably want to take out the tumor.
So we have to stop here. I know there are many good questions.
I'll stay around and
you want to ask. The lesson I learned, the two biggest things that I need to do to avoid cancer is, one, not smoking. Second, not aging.
There you go.
By the way, there's a very good book. Some of the questions asked, I didn't want to interrupt Professor Nadelli's lecture. There's a very good book about history of cancer called, I'm sure you know this.
The Emperor of All Maladies.
Right.
The author is Mukherjee, and I actually set it out today. I may have brought it. This is such a wonderful book. It chronicles cancer from back at the time of the Egyptians. I do happen to have it here. Wonderful. This is really an extraordinary book, and it not only tells you about developments of cancer, it's wonderfully written. It received the Pulitzer Prize in 2011. So it's not just extraordinarily informative, but it is wonderfully written. It's a terrific book.
Two-thirds through of this book. So, some of the questions actually people ask, for example, early detection. That's what really happens. It's a philosophical question, right? Now we have more cancer cases. Maybe it's due to the technology that we can detect better. But what is the way, the most important thing that can really prevent or improve people's life. It's not really sort of treatment or remission or time of the remission that matters because if you can detect early, then you can claim that people can live longer after they've detected cancer. So read that book. I'm not going to tell you the conclusion, but I do want to thank Professor Nadali for a nice, very nice